So it's my pleasure to have Davide Bolognini here today. Davide has got his PhD from the University of Siena, and afterwards he has worked in several institutions in Italy and in Europe. Now he is a senior bioinformatician at the Human Technopole, so it's close to us in Milano, and is going to talk about the inference of complex haplotypes from sequencing in a pangenomic settings. So, Davide, thanks again for having accepted our invitation, and please go with it.
Yeah, I hope you can hear me well. So, yeah, good morning, everyone. And first off, I'd like to thank the organizers for having me here. So, my name is Davide, as Gianluca was mentioning. I'm a senior member of the Genome Analysis Unit, which is a unit within the Population and Medical Genomics Research Center at the Human Technopole in Milan. We offer computational support to groups both within and across centers at the Technopole. And beside the computational support activity, we are also involved in several research projects. So more specifically, I'm the one dedicated to projects involving long-read sequencing and pangenome-related stuff, and that's why I'm here. Today, I'm gonna talk about the topic that you see here in the title. Sorry, so I have a different screen for the presentation. That is approaches in general that explore the pangenome idea to improve on genotyping. So the first part is a bit didactical, so be with me for that. I have no idea of how the others tackled the presentations problem, but yeah, so I will be very quick on the first part, and then I'll go on with a couple of approaches that I think are interesting. So in general, what is genotyping? Diploid organisms have two copies of each autosomal chromosome, each of which can carry genetic variation. So the process of determining whether a certain variant allele is located on none, one, or both of these copies is called genotyping. So for instance, here you have the schema of a sample with two haplotypes. And I have highlighted three alleles, which are the green one, which is present in both haplotypes, the yellow and the blue ones, which are present just in one. So routinely used genotyping methods for sequencing data are based, for instance, on short-read alignments to the reference genome. And I have highlighted here the alleles that the reference genome exhibits. So you have the red allele, I hope you can see my pointer, and the blue allele here. Yeah. So and this what you see here is the possible alignment of the sample reads to the reference. Yeah. So in the first block, there is no variation highlighted. So you would say that in that region, the sample haplotypes are both equal to the reference. So we can call RR. In the second block, the reference is red, but our sample only has green reads. So we infer that both the haplotypes are alternate. So AA. Again, a third block, which is equal to the reference. And then the fourth block is blue in the reference, but there is a mix of yellow and blue in the sample. So we derive that one haplotype is reference and the other one is alternate. And again, a final block, which is equal to the reference. So again, reference. So this is usually represented in the VCF specification with the following entries. Yeah. So you have 0, 0, 1, 1 for alternate, and so on. So this is the routine way to do genotyping from short reads. And this is the best case scenario where you still are able to somehow identify the genotype of an individual, even if not all the alleles are present in the reference. Yeah? These genotyping methods, however, suffer from the so-called reference bias, so which occurs when reads originating from non-reference alleles fail to align to the true point of origin missing in the reference, or those reads aligned to a different locus. So for instance, here the yellow reads fail to align, and they are left unmapped here, like on the right side of this slide, because the yellow region is not represented in the reference genome, and this causes wrong genotyping results in effect. So you may infer that here the sample is reference, what in effect he is not. So one possibility would indeed be to use many references that can better model the global variation. So for instance, here, in addition to the usual reference, you have three other haplotypes that are ordered from 1 to n that exhibits all the alleles in the sample we want to genotype. While the first, third, and fifth block here are shared, so those that are highlighted in light gray, let's say. The red allele, the blue and the yellow one are present only in certain haplotypes. And reads here can map without ambiguity. Of course, so the effort of generating these high quality assemblies that then can be included in larger reference panels is the aim of several consortia, including the AGSBC, which is the Human Genome Structural Biome Consortium, and the HPRC, which is the Human Pangenome Reference Consortium. So AGSBC latest release contains like 65 deployed telomere-to-telomere samples, while HPRC released 47 assembled individuals in the year one release, and now there are more than 200 individuals. So generating these high quality assemblies is somehow still not that cost effective. So that's part of the reason why still linear reference-based approaches are used. And we as Technopole have a partnership with the HPRC, and we have just finished the sequencing and the assembly of 4 TSI individuals, which are basically people from Tuscany, and those will be included as an Italian representation in the next Pangenome release. So we are quite proud of that. Okay, so there are a couple of methods out there that can be used to genotype individuals, so solve the problem that we presented now using a collection of assemblies, for instance, in the form of a pangenome graph. I won't go through all of them, of course, so I talk about 2. So the first one is Pangini from Jana Ebler and the Masha group. And the other one is COSICT, which we developed at the Technopole together with Eric Garrison and Peter Studman's group. Of course, since I'm not the developer of Pangini itself, I may miss some bits, but I find the approach really interesting, and that's why I'm bringing this here, again, for like didactic purposes. Okay, that's the paper. So everything I will tell you about this method we also find in the paper. I've just tried to summarize it like with sketches so that it's easier for everyone to remember. So Pangini relies on a pangenome built from a collection of variants in BCF format. That's one way to represent pangenome. So I'm not sure if you already went through methods for pangenome construction, probably yes, but it is just one way to do that. So in particular, in the paper, they used haplotype resolved assemblies for 14 samples. So those, for instance, that you see here. In practice, for each sample, you can separately map contigs of each haplotype to the reference genome. So in that paper, they used GRCH38. So what you see here with R. This can be done using standard aligners, such as Minimac2, and then they call variants. So you have the first haplotype, you align against the reference, and then you do variant coding. And for instance, here you see that haplotype 1 and reference differs for a certain block, which is the green one here, which is red in the reference. You do the same for the second haplotype, and you see an additional bit of variation. And then so you do the same until you align all the haplotypes in the collection. And then you can use these variants to construct the graph. In particular, the graph they construct is acyclic and directed, and it is constructed by progressively inserting the variants of all haplotypes into the linear reference genome. So basically, you start by inserting the green block here, and you generate this bubble. Then you add the yellow part, which results in an additional bubble, and then you add variants from the HN haplotype, which result again in a more complex bubble. Yeah. So variants produce bubbles in the graph with branches that define the alleles. The haplotypes, of course, are passed through the resulting pangenome. Yeah. So what then Pangini does, so for each bubble, it determines a set of k-mesh that are uniquely characterize the bubble itself. So this is done by counting all the k-mesh along the haplotype paths and determining then a set of k-mesh for each bubble that occurs at most once within the variant bubble itself. So that's for just an example. So you will have, for instance, 4 unique k-mesh for the green part of the bubble, 3 for the light blue one, 2 for the red, and so on and so forth. Okay, so once we have the setting here, so we have the k-mesh from each bubble, and then you get a sample that you want to sequence from which you get short reads. In practice, what they do, so they count the k-mesh from unique bubbles into the sample reads and integrate this in a more complex model that we will see in the next slide. So basically, you have the k-mesh from unique branches of the bubbles, and you count this in the reads. So for instance, this k-mesh here is seen in 9 reads. This other k-mesh here is not seen at all, and this makes sense because this part is not present in this specific sample here. Again, for the yellow, this is seen in 4 reads, and for the dark blue one, so it's seen in 3 reads. From this, you can derive copy number state. So very naively, this can be done by just dividing the number of reads that you get by the average number of reads for each k-mesh that you see. So basically here it will normalize to 2, to 1, and to 1 here, and 0 because this is not seen, for instance. Okay. So the distance, so the number of k-mesh that you see and the distance between bubbles, those are informations that are integrated into a model, which is an HMM, and these models the unknown genome as a mosaic of the provided haplotypes, yeah, and reconstruct it based on the k-mesh counts observed in the sample sequencing reads. So because you observe these counts here, the genotype will of course be green for the first block and yellow-blue for the second block. And the distance, as I was mentioning before, between bubbles is also taken into account in the hidden Markov model. That is somehow influences the transition probabilities of the model itself. And this is also, of course, useful to genotype bubbles that exhibit some uncertainty. For instance, here, in the first bubble, k-mesh count suggests that the sample probably carries the alleles of the green and the blue haplotypes, while in the second bubble, because this is poorly covered by k-mesh, you may have some uncertainty. But because this bubble here is linked to the previous bubble, this can be used to infer the 2 local haplotypes path for the second bubble, yeah? And it will likely result in the green-blue path inferred from the previous bubble genotype, yeah? Which is, yeah, I mean, I think kind of a pretty cool approach. It has some limitation, of course, especially in scalability. But the interesting part, I think, it's the approach itself. Yeah? There is another tool which is Lossy-Typer from the same group. I'm not going into details of this as well. I'm just leaving this here in case anyone wants to give it a read. So in Pangini, they use a sort of augmented sequence graph model. So you start from the reference and progressively add variation. Yeah. So we have developed our own method of genotyping from graph. This is called COSIC, that stands for cosine similarity based genotype. In COSIC, we use the variation graph model, which describes the all-to-all alignment of many sequences as works to a graph whose nodes are labeled with DNA sequences. So let's just try and build a graph from these three samples here. So we have six sequences, right? So in this, can be considered a pangenome itself, because pangenomes are basically just a collection of sequences, but we can use these sequences to build a variation graph. So the first block, which is the black block, is shared across all the haplotypes, and you will have a node. And then from the black one, you can go through to the red or to the green node. So you will have either, so you can take the red path or directly the green one, or go through the red one and then to the green. And then you have blue and purple, or I don't know what kind of color is this. But there's also one haplotype that loops twice over the blue node. So this is why you see a double loop over this node. And then again, you can go through the yellow or the pink node, and then again to the gray one. That's how you construct the variation graph from these six paths. Yeah, and of course those are nodes. So those here are represented one linked to the others, but of course those are nodes, and there are edges connecting nodes. So if you take there are a couple of concepts that are useful to understand the approach that we use. So the first one is defining the so-called path coverage vector. So if you take the node from the graph and you plot like this, there's just a collection of all the possible nodes in the graph, and then you can count the number of times each path crosses each node in the graph. So for instance, for these two paths here, you will have something like this. So the first path crosses the black node once, the red node once, again once for the green node, once for the blue node, once for the pinkish node, once for the yellow, 0 for the pink, and 1 for the just because the pink node is not present in the path. Yeah, and you can do the same for the second haplotype. So you will have coverage 1 for the black node, 0 for the red, 0 for the yellow, and 1 for all the others. So this is the so-called path coverage vector. So the number of time each path crosses a certain node in the graph. And it's node space because it takes into account all the possible nodes, because of course some paths may not have certain nodes. Once you have a sample, for instance, those are reads that are generated from a sample, you can take the reads and align those reads to the nodes in the graph, because nodes are sequences, so you can just take the reads and align to the nodes and derive a sample coverage vector, which is just the number of reads aligning to each node in the graph itself. And that's exactly what I was referring to. So for instance, you have 24 reads here, 12 reads in the red node, and so on and so forth. For all the others, you see numbers here. Okay, and these three concepts here, so the variation graph, the path coverage vector, and the sample coverage vector, are everything that we need to try and genotype a sequence sample using a pangenograph, using the approach that we built. So that's the workflow. The thing that you see here is on the left is the so-called rule graph. So the rule graph is like tells you where you come from, where you go when you specify a certain rule, and this comes directly out from one of these 2-3 workflow languages, which is SnakeMake, and indeed the workflow itself is implemented in SnakeMake. We are going to break this workflow down into pieces, so don't worry if it looks like confusing at first. So let's say that we want to genotype a certain region in an individual. What we do can be summarized more or less in 4 steps. So the first step is to build a graph for the region. So that's a region-specific graph, because working with graphs that are too large becomes impractical. So the first stuff that we do is to pinpoint to the region we are interested in, and then we build locally a variation graph for that part. And then we compute the sample coverage, what I was mentioning before in the introduction. We compute the path coverage, again, one of the previous slides. And then we do the so-called haplotype deconvolution, which is the genotyping itself. So at the moment, our workflow requires as inputs, so chromosome level assemblies like those that are generated from like HESVC, HPRC, or whatever, just those that we know like belong to a certain chromosome, reads from one or more individual and one or more region of interest that we want to genotype. And the output are like the sample haplotypes, so the 2 haplotypes in the graph that best describe the sample, and the structural clusters for these haplotypes. But yeah, we will see this like in a couple of minutes. So the cool thing of having everything implemented in a workflow language is that it's easy to genotype in parallel. That is, we can like do what I'm describing now for a single region across many regions and many samples at the same time, which is pretty convenient if we want to scale at biobank scale or like many samples, yeah? And many regions. Okay, so that's as I said, we have like 4 main steps, which are those that I'm going to like explain a bit better. And so what you see here in gray and black is the reference, and we refer to these as target. And the other assemblies, we usually call them queries. Yeah. So the first thing that we do is a query to target alignment. And then we leverage the so-called implicit pangenome graph model to extract the region of interest. So basically, we have the queries aligned to the target, and we can use so the implicit pangenome graph leverages COI trees, which are implicit interval trees, to provide like efficient range lookup over the input alignment. So we have the queries aligned to target. We know the coordinate of the region of interest in the target. For instance, a certain region of GRCH38. We can use the pangenome, the implicit pangenome graph model, and the COI trees to project the region of interest from the target into the queries. Yeah. So once we have the queries aligned to the target, so we project the region of interest into all the assemblies. And once we have the coordinates of the region of interest on all the other assemblies, we can just pick these regions from the assemblies and use them to build a variation graph. This is done using PGGB that has been developed by Garrison and Guadracino. But yeah, in effect, and this allows us to build region-specific graph. And for instance, here you have the C4, which is the complement for region on human chromosome 6, which has these aspects here. This is just a sketch, of course. The true aspect is like this one here that you see on the left. So once we have the region specific graph, we can compute the path coverage. And we said that the path coverage is just the number of time each path crosses a node in the graph. So what you see here is basically the path coverage for two different paths. So the first path is haplotype 1 from this individual here, AG00096. And the graph that you see, the plot that you see in the bottom part of the slide is the second haplotype of the same individual, right? So each point here is a node. What you see on the x-axis is the cumulative length of all the nodes that are in the graph, and you see that some nodes have coverage 2, which means that path crosses that node twice. Some others have 1, and some others have 0 because those nodes are not represented in that path. That's the same for the second haplotype of the AG0096 sample. So some nodes have 1, some nodes have 0. You don't have nodes with 2 coverage, for instance, here. Just another example for another sample, this AG00171. So you see that some nodes have 0, some nodes have 1, 2, and there are just a couple of nodes that coverage 3 and 4. Yeah, which basically means that that path crosses that node 4 times or 3 times with respect to the height of the point that you see here in the plot. And same here for the second haplotype. Yeah. Okay, so because we want to genotype a certain sample, so we take his weights and we map them to the regions that we have extracted from the assemblies. And this can be done using standard aligners. We use BWA-MEM for this. We use actually BWA-MEM2, which is like a bit more fast. But in practice, you can ideally think about extending this to Minimap2 with long width presetting if your input are reads coming from Oxford output technology sequences, for instance, or PacBio sequences. And once we have the reads mapped to the blocks that we have extracted from assemblies, we can project back the coverage that we have for each block into the graph. And what we have, it's a sample coverage vector. So if you just look at the upper plot, each point again here is a node. And if you look at what you have on the y-axis on the left, yeah, that's the sample coverage. So for instance, here the sample has coverage 100, here sample has coverage 40, more or less. And this can be overlapped with the pangenome coverage vector. So for instance, here you have reads coming from AG0096 sample that's been sequenced with short reads. And below you have the pangenome coverage for the 2 parts of the same individual overlapped with a sample coverage. Yeah? So the 2 y-axis have different scales because of course one is from the sample and the other one is from the pangenome, right? You can of course overlap these to other haplotypes. So this is, for instance, the same sample, but the pangenome coverage represented here comes from haplotypes coming from a different sample, so AG00171. So once we have this setting here, so we have the path coverage vector and we have the sample coverage vector. So the question that we want to address is the haplotype deconvolution itself, which is the proper genotyping step. So the haplotype deconvolution problem can be break down to find the combination of paths best representing the trend observed in the coverage vector. And of course, the combination will be of two parts because we are dealing with diploid samples for human, and each part is haploid because it's like a single haplotype, and we have two, so we need to combine two parts in the graph and like check which is the combination that best fits with the sample coverage vector. And that's why n equal to here for the diploid sample. What we do in practice is to compute. So this can be done in many different ways. We chose to use the cosine similarity for that. So the cosine similarity is basically the angle between two vectors. Is calculated. So having two vectors is calculated as the dot product divided by their norm. And in practice, what we do is schematize as follows. So we have the sample coverage and we have the path coverage vectors. So what we have highlighted like in the previous slides, we generate all the combination of path coverage vector of two. This includes, of course, for heterozygous and homozygous combinations. So you will have for these three H1 plus H2, H1 plus H3, H2 plus H3, but also H1 plus H1, H2 plus H2, and so on and so forth. And then we calculate the cosine similarity of the sample with respect to all the possible haplotype combinations. And then we pick the combination with the highest cosine similarity score. So for instance, here, this sample coverage vector is best described by the combination of H1 and H2 because the cosine similarity here is the highest. Yeah. One thing that is actually true is that many haplotypes are structurally similar, and that's something that you can also see by eye. So in this plot, actually the haplotypes are already sorted by similarity, but each line here is an haplotype. And what you see is that many haplotypes actually are very, very similar. For instance, you have a first block, let's say this is group 1, with haplotypes that they look like very similar, right? Because the red part is where the path coverage increases. So you will have some regions that are gray, which means that they have one in terms of path coverage, and then you have some regions that are red, which means that they have two or more in terms of path coverage for the nodes that are highlighted in these regions. Yeah? And then you have another group again. So group 1, group 3, group 2, group 3. So all these haplotypes here, in terms of large variations, are very similar. Yeah, and then you can define at the same time group 4, group 5, group 6, and group 7. So what we found was that it kind of makes sense not to just output the best combination of haplotypes that describe a certain sample, but also define the structural cluster of each haplotype so that we can predict not just a single combination, but the haplotype cluster each haplotype belongs to. At the moment, we are doing this using just agglomerative clustering with a silhouette analysis for defining the best number of clusters, and then once we have the number of clusters, we cut the dendrogram tree, and we use that many clusters that it comes out from the silhouette analysis. But in practice, this makes a lot of sense also from a gene structure point of view. So what you see here are clusters with gene injected. Like those are gene annotations that are injected in the graph, and haplotypes from the same group here are very similar structures. For instance, all haplotypes in group 2 here miss, for instance, C4A. So the region here is the C4 on chromosome 6, which is characterized by C4A, C4B, and some other genes that are not that important at the moment, but all haplotypes, for instance, in group 2 miss C4A, but also some other bits. while those in group 3 have, for instance, longer C4b with respect to those in group 1. That's just an example. By like predicting a couple of haplotypes, maybe like you may be able to predict a couple of haplotypes that falls in the proper cluster without predicting the proper one. So let's say that we have a sample like it's AG00096 or whatever. We may not be able to predict exactly haplotype 1 and haplotype 2 from AG0096, but we are still able to predict clusters from the proper clusters from the respective haplotypes. Yeah. So that's just an example of benchmarking that we do. So we have HGSBC version 3, for instance, 65 samples. From these 65 samples, we have 64 matching short reads that are coming from 1,000 genome. So we take a sample, we genotype. So we take a sample that has been sequenced, we genotype with respect to the graph, and we assign both the couple of haplotypes and the structural cluster to each individual, right? And then how do we check if our predictions are correct? So we ask basically 2 questions. So if the sample has been assigned the proper haplotypes, we expect it to be S1, S2, because they are in the graph, so we are able to see if those are like correct. Or if we have a sample that is being assigned the proper clusters for the two haplotypes. Yeah? In that case, it would be a true positive. Otherwise, it's an error. And for this region here, we have 63 out of 64 of these samples that are properly genotyped. So this basically means that our accuracy is pretty high, at least for this region here. Of course, we can repeat this by like removing one haplotype from the original graph and try to like regenotype the sample, checking just for cluster, and this will be called leave one out. But yeah, we can talk about this later. I mean, just in the interest of time, I'm gonna skip this part. Yeah, okay, so that's more or less how it works. We are like currently experimenting with like refining this approach. So one thing we want to like do is to explore if like removing nodes that have stable coverage across all the paths somehow helps like improving the genotyping itself and the clustering, because of course the nodes that have stable coverage across all the paths do not contribute to variation, and therefore should not be like have a big weight like when clustering, right? And then another thing that we are doing is like not using agglomerative clustering, but instead switching to dbScan so that we can better control the intra-cluster similarity of the elements that goes within each haplotype cluster. And at the moment, we are also extending this to many other loci, and the idea will be to run this across at least 100 to 100 loci before having a stable release that we can publish. But okay, so of course this is not just work of the Technopole, but we have many collaborations with people, especially in US. Eric Garrison and Andrea Guarnacino here, and Peter Sutman and the group is Jason Chin that really helped us a lot, especially in the first part of the development. So this approach has already been published in Nature as part of this paper here that's focused on the amylase. So basically what I showed for C4 locus, this has been applied to amylase. What you see here on the right are the structural clusters of amylase as they were defined by the approach that are described before. And I'm also leaving this here if you have like if you want to give it a read or like curious about how this can be applied to different data sets, because here we didn't just use it on modern short read samples, but also on ancient DNA samples, and it works pretty well also at very low coverage levels. Yeah, and that's it from my side.
Thank you very much. It was very clear. And we are waiting for questions from the audience. Again, I remind everyone to write your questions on the WhatsApp channel. One quick question from me. Uh, what are the computational requirements? So,
Yeah, yeah, no, no,
how long did it take? Because I mean, it's impressive what you have done, but I want to see how easy it is to replicate.
yeah. Okay, so I guess it really that so this is like more convenient to run if you have many regions that fall within the same chromosome, right? Because let's say that you want to genotype 100 regions that are on chromosome 6. So the very first part, so the alignment where you have all the queries to target alignment, is done just once per chromosome. So let's say that you have all the contigs from chromosome 6, so you align all the queries to chromosome 6. And then once you have that part, right, it's pretty fast, because so once you have the alignment of all the queries to the chromosome, then like slicing and identifying the single blocks inside the chromosome is super fast. And also the local graph construction, which otherwise will take like days, right? Because building a graph for an entire chromosome is too time consuming. But because we are doing this locally and in parallel, then it's super fast. So for instance, like I don't know if you have for amylase locus, just one locus, but we genotype this across thousands of samples, and it took like 3 or 4 hours. Yeah, so that's
3 or 4 hours, but on a huge server, on a
it's just yeah, it's just like one computation. Yeah, it's like 32 nodes, but just each node using one CPU. So so it's like, well, you can't actually think of parallelizing this without efficiently parallelizing this without having a cluster. That's you need to rely more or less on a cluster for efficient parallelization. But yeah, that's what I mean. I mean, if you have like some computational power, like 32 CPUs, for instance, then it's like super fast. So the efficient the part that's more that takes the more time is, as I was mentioning here, the alignment of queries to targets, which is like a bit time consuming. It's like alignment, but all the other stuff, it's very local, so it's like reduced to the minimum terms.
and is the software available?
Yeah, exactly. So I think I have like the
You will
link
post it.
here. Yes, exactly. So it's
Okay, okay,
yeah, it's also present in the slides.
okay. Let's see. So I see no questions in the chat. Let's wait a few seconds if someone asks there. Otherwise, I think that we can thank again Davide. Very, very nice talk. I enjoyed it a lot. And thanks again for being here.
Thank
So
you.
it was a pleasure.